Hey everyone, it's 11 o'clock here in Minneapolis, Minnesota, so we're going to get started with our webinar today. Thank you all for joining us today. I'm Sally Head-Dahlquist, President here at Immunochemistry Technologies, and I'll be moderating the webinar today along with our Marketing Associate, Allison Westcott. You may have noticed that our phone has been muted. However, we encourage you to ask questions along the way. Simply type your question into the chat dialog box at the right and select Send to Host. If you have any technical difficulties during the webinar, please send your chat to me and I'll do my best to get you up and running. We will be posting the video recording of this webinar later this afternoon or tomorrow, so look out for an email from us after the webinar with a link to the recording, as well as a discount code for some of ICT's oxidative stress products. Now I'd like to introduce our speaker, Dr. Christy Strandberg. Dr. Strandberg is a staff scientist here at ICT. Her primary responsibilities include research and development, mainly focusing on the development of new targeted probes and intracellular fluorescent reagents. She also provides technical support, so if you need help with an assay, she may take your call. Prior to joining ICT and moving back home to Minnesota, Dr. Strandberg worked on analytical method development and validation at Emergent Biosolutions in Michigan, and before that, she worked as a postdoc researcher at The Ohio State University in their Center for Microbial Interface Biology. Dr. Strandberg earned her PhD in 2009 from the University of Minnesota's PhD program in microbiology, immunology, and cancer biology. We are honored to have her here with us today as our presenter. Today, Dr. Strandberg will be discussing oxidative stress. Immunochemistry Technologies offers multiple tools for the detection of oxidative stress, including a line of in vitro cell-based assays that measure changes in the levels of reactive oxygen species and nitric oxide synthase in living cells. ICT also carries a line of products for detection and quantitation of extracellular reactive oxygen species. In this presentation, learn how to detect the presence of oxidatively damaged DNA, quantify levels of reactive oxygen species, such as hydrogen peroxide and nitric oxide, as well as how to monitor mitochondrial membrane potential. Ultimately, these assays enable researchers to obtain a better understanding of the oxidative stress-associated events that occur as a result of drug treatments, various disease states, and other experimental conditions. And with that, I'll turn it over to Dr. Christy Strandberg.
Thank you, Sally, for that lovely introduction. So before I dive into today's webinar topic, I want to take a quick moment to tell our listeners a little bit about Immunochemistry Technologies. We are a woman-owned and operated company that was incorporated back in 1994, so we've been around for quite some time now. We began as a company that provided custom assay services. Now, this includes activities like immunoassay design and development, antibody purification and conjugation, and so on. I do want to mention that we continue to offer our custom assay services. A few years ago, we expanded our company to offer lines of consumable reagents as well. This includes ELISA solutions such as coding buffers, blockers, diluents, and so on. We also offer a line of projects, as Sally mentioned, that are tailored for detecting cell health status. Included in this is ICT's line of products for detecting oxidative stress, which will be the primary focus of today's webinar. With our expertise in ELISA development, we do offer a variety of immunoassay-related services. Our scientists have years of experience with protein chemistry and ELISA optimization, so we can help you develop reliable, sensitive, and specific immunoassays. We can also scale up and manufacture an assay for internal use for our clients. If you're in need of a service project, feel free to reach out to us. We'd be more than happy to discuss your project with you. Before moving on, I do want to briefly touch on our panel of ELISA solutions. These include all the components you need to build a better ELISA. Our coding buffers, blockers, sample and assay diluents, conjugate stabilizers, and wash buffer all work together to minimize the buildup of unwanted proteins. These products work together to address common issues associated with specificity, sensitivity, reproducibility, and shelf life. Our ultimate goal is to help you develop an optimized ELISA that has a high specific signal and low background noise. That's all I'll mention about our ELISA products today, but if you do have questions about this product line, feel free to chat them in and I can get you a response after the webinar. I also want to mention that ICT offers a variety of fluorescent probes. In addition to the oxidative stress products I'll be discussing in today's webinar, we also carry numerous products for the detection of caspases, cathepsins, and serine proteases, as well as other auxiliary stains like propydium iodide and hooks. If you have any questions about the other products or services offered by ICT, please contact us by emailing help@immunochemistry.com or by calling us at 1-800-829-3194. So now on to the topic of oxidative stress. I want to begin by discussing some of what I consider to be important background information. The first concept I want to introduce is that of the oxidation-reduction reaction, which is otherwise known as a redox reaction. This is a type of chemical reaction that involves the transfer of electrons between two species. During a redox reaction, the substance, be it a molecule, atom, or ion, changes by either losing an electron, in which case the molecule has been oxidized, or by gaining an electron, which has the end effect of reducing the amount of positive charge, in which case the molecule is said to have been reduced. Disturbances in the normal redox state of cells can lead to the buildup of toxic molecules called reactive oxygen species. These molecules are capable of damaging DNA, lipids, and proteins. Reactive oxygen species, or ROS, are chemically reactive molecules containing oxygen. From a biological perspective, reactive oxygen and nitrogen species are formed as a natural byproduct of the normal metabolism of oxygen. One such example of ROS production takes place in the mitochondria, which are the organelles responsible for generation of most of a cell's energy. Mitochondria produce reactive oxygen species called superoxide as a byproduct of their normal activity. The superoxide that leaks out from the mitochondria is converted into hydrogen peroxide, and under normal conditions, the detrimental effects of hydrogen peroxide and superoxide are ameliorated by enzymes such as catalase and superoxide dismutase, which break down these damaging molecules into oxygen and water. ROS play important roles in cell signaling and homeostasis. Their production can increase dramatically during times of environmental stress. It's also important to note that these molecules possess antimicrobial properties and are involved in the immune response against invading pathogens. Some examples of reactive oxygen and reactive nitrogen species include superoxide, hydrogen peroxide, hydroperoxide, hydroxyl radical, and peroxynitrite. Now, this slide shows a simplified, but I'll say still somewhat detailed, schematic depicting the formation and decomposition of reactive oxygen species. I've included this slide just to give our listeners a better appreciation for the complexity of cellular redox reactions. I won't go into much detail here. I just mainly want to share this figure to illustrate how complex this system is, and to highlight that there are many different enzymes and molecules involved in the formation and decomposition of ROS. For example, let's consider the formation of superoxide. The reduction of molecular oxygen produces superoxide. This molecule serves as a precursor to most other reactive oxygen species. Dismutation of superoxide produces hydrogen peroxide, as I mentioned before in that mitochondrial example. Hydrogen peroxide can then, in turn, be partially reduced to form a hydroxyl radical or fully reduced to form water. Under normal physiological conditions, cells control ROS levels by balancing the generation of ROS with their elimination by scavenging molecules such as antioxidants. However, under conditions of oxidative stress, excessive ROS can build up and cause damage to various cellular components, such as DNA, lipids, and proteins. Most of the harmful effects of reactive oxygen species exposure are the damage of DNA, the oxidation of polyunsaturated fatty acids and lipids, this is also known as lipid peroxidation, oxidation of the amino acids found in proteins, and the oxidative deactivation of specific enzymes. As I just mentioned, the buildup of reactive oxygen species can have numerous effects. One such effect is that ultimately oxidative stress can trigger cell death. Moderate levels of oxidative stress can cause a cell to initiate apoptosis, which is a programmed form of cell death, while severe levels can trigger necrosis. Here I'm showing a schematic that depicts apoptosis occurring through two different pathways, the intrinsic pathway and the extrinsic pathway. If you'd like to learn more about cell death and apoptosis, as well as our products for detecting cell death, please visit our website. I've given a couple webinars where I go into greater detail on this topic and on necrosis and autophagy as well. You can access these resources from our webinar library. On the mention of autophagy, one last comment I want to make on this topic of cell death does pertain to autophagy. Some recent studies have suggested a link between ROS levels and autophagy. This association is thought to occur as the cell tries to rid itself of harmful, damaged organelles. However, I do want to mention that there's a lot yet to be discovered on this topic, so I won't be going into that one in any detail today. So an imbalance in the production of reactive oxygen and nitrogen species is associated with many different disease states or pathologies. As I just mentioned, an overproduction of free radicals can result in the damage of proteins, DNA, and lipids. The simple cartoon shown on this slide portrays lipid peroxidation. The overproduction of reactive oxygen species has been associated with conditions such as atherosclerosis, diabetes, cardiovascular disease, cancer, and chronic inflammation. The link between cancer and chronic inflammation is more complex than it may appear on the surface. Chronic inflammation, which in part is regulated by ROS, is considered to be a major mediator of cancer. Interestingly, modest levels of ROS are required for cancer cells to survive, whereas excessive levels of ROS can kill cancer cells. So the take-home message here is that the overproduction of reactive oxygen is involved in many disease states. However, the production and elimination of ROS in biological systems can be complex, to say the least. So much of this presentation thus far has focused on the damaging effects the production of reactive oxygen and nitrogen species have on the host. But it's important to remember that these molecules serve beneficial functions within the organism as well. I've touched briefly on how they serve as signaling molecules and are involved in homeostasis. But also in mammals, reactive oxygen species are produced as an antimicrobial defense during infection. Cells such as macrophages and neutrophils are just a couple examples of cells that produce ROS. Exposure to ROS can be detrimental to a pathogen, for example, by causing DNA damage, which ultimately can kill the microbe. The importance of such a defense is evident in individuals who have deficiencies in generating ROS. One such example is chronic granulomatous disease. These individuals are highly susceptible to infections that are caused by a broad range of microorganisms, such as Salmonella enterica, Staphylococcus aureus, Serratia marcescens, and Aspergillus. Switching gears, I want to begin introducing ICT's variety of products for detecting oxidative stress. One of our most popular oxidative stress kits is the Intracellular Total ROS Activity Assay. The Total ROS kit utilizes a proprietary oxidation-sensitive probe called Total ROS Green. On its own, this cell permeant dye is non-fluorescent, but when in the presence of intracellular reactive oxygen species, this dye becomes oxidized to its green fluorescence-capable form, making it easy to detect by common methods such as flow cytometry, which is the data I'm showing you here on this slide. These histograms depict the flow cytometry results from Jurkat suspension cells that were preloaded with the Total ROS Green dye and then were exposed to 100 micromolar tert-butyl hydroperoxide for 30 minutes to induce oxidative stress. The increase in ROS activity is evident in the overlay histogram, which is shown at the far right. The MOC-treated cells are represented by the black line, and the tert-butyl hydroperoxide-treated cells are represented by the red line. Here you can see a nice shift in median fluorescence intensity. Hydrogen peroxide is a molecule of great interest. In biological systems, the one electron or incomplete reduction of molecular oxygen produces the highly reactive superoxide anion radical. Further reduction of the superoxide radical by the enzyme superoxide dismutase results in the creation of hydrogen peroxide. Hydrogen peroxide is a relatively stable oxygen form which, unlike its more oxidized precursor, is capable of readily penetrating cell membrane barriers and influencing intracellular metabolic activities. Hydrogen peroxide has important roles as a signaling molecule in a variety of biological processes. For example, hydrogen peroxide is thought to serve as a signal to immune cells to converge on a site and initiate defense and promote healing. ICT offers both a fluorescent and a colorimetric hydrogen peroxide detection kit. These kits are available in a 96-well format and are designed to quantitate hydrogen peroxide in a variety of samples, such as fresh urine, buffers, and tissue culture medium. The graph shown here depicts the standard curve from a typical hydrogen peroxide fluorescence detection kit. In this kit, a hydrogen peroxide standard is provided to generate a standard curve. Samples are mixed with the fluorescent substrate, and the reaction is then initiated by the addition of horseradish peroxidase. Samples are then incubated at room temperature for about 15 minutes, and any hydrogen peroxide present in the sample will oxidize the horseradish peroxidase, which in turn oxidizes the colorless non-fluorescent substrate, converting it into a fluorescence-capable product. Increasing concentration levels of hydrogen peroxide cause a corresponding increase in the horseradish peroxidase oxidation rates, which translate into a linear increase in the amount of fluorescence product detected. As I mentioned before, DNA is one of the critical molecules that can be damaged during times of oxidative stress. Hydroxylation of the guanosine found in DNA occurs in response to both normal metabolic processes as well as a variety of environmental factors, i.e., anything that increases reactive oxygen and nitrogen species production. Hydroxylation of guanosine is depicted in the diagram shown to the right. 8-hydroxy-2-deoxyguanosine, or 8-OHDG, is produced by the oxidative damage of DNA and serves as a well-established marker for oxidative stress. Increased levels of 8-OHDG are associated with the aging process, as well as with a number of pathological conditions, including cancer, diabetes, and hypertension. Oxidation of DNA can cause many types of DNA damage, including single-strand breaks, protein DNA adducts, as well as intra- and interstrand cross-linking. ICT offers a competitive ELISA for the quantitation of 8-hydroxy-2-deoxyguanosine. Our kit is compatible with a variety of sample types, including urine, cell culture medium, plasma, saliva, and tissue samples. This assay utilizes an 8-hydroxy-2-deoxyguanosine-coated plate paired with an HRP conjugated detection antibody that's specific for 8-OHDG. A typical standard curve is shown in the graph displayed on this slide. As this is a competitive ELISA, the greater the concentration of 8-hydroxy-2-deoxyguanosine in the sample, the lower the end signal detected by the assay. How this works is that the greater amount of 8-OHDG in the sample, the more likely the detection antibody will bind to the free 8-OHDG present in that sample instead of binding to the 8-OHDG coated on the plate. Only the detection antibody that is bound to the 8-OHDG coated on the plate will be retained and detected. Antibody that is bound free 8-OHDG will be washed away. So oxidative stress can also affect mitochondria. The mitochondrion is a double membrane-bound eukaryotic organelle found in most cell types. The inner membrane of the mitochondrion is where cellular respiration takes place. This process is responsible for generating most of a cell's energy in the form of ATP, or adenosine triphosphate. Mitochondria produce energy by establishing an electrochemical proton motive force across their inner cell membrane, which fuels the synthesis of ATP. The electron transport chain is responsible for creating this electrochemical proton gradient. This slide shows how mitochondria produce energy by establishing that proton motive force I mentioned. And that, like I said, that's what fuels the synthesis of ATP. ICT offers several potentiometric dyes that can be used to assess the status of this proton gradient. These dyes are called TMRE, TMRM, and JC1. The image shown on this slide depicts the accumulation of TMRM, which is shown as the red chemical structure that appears to be a darker shade of red when accumulated in the mitochondrion. Inside a healthy cell, this positively charged lipophilic dye enters the negatively charged mitochondria, where it accumulates and then fluoresces upon excitation. Oxidative stress can trigger the depolarization of the inner mitochondrial membrane, resulting in dissipation of the protons that had accumulated within the intermembrane space. Depolarized or inactive mitochondria have decreased membrane potential and fail to sequester dyes such as TMRM, resulting in a measurable drop in fluorescence signal, which can be easily measured by flow cytometry, fluorescence microscopy, and so on. This slide shows a measurable drop in fluorescence signal as mitochondrial inner membranes depolarize. Here we're looking at some sample flow cytometry data showing TMRM staining and microscopy data showing TMRE staining. Jurkat cells were treated with 1 micromolar staurosporin for 3 hours to induce apoptosis, and then were stained with either TMRE or TMRM, respectively. The untreated samples are, for the flow histogram on top, the untreated samples are shown as the red peak, and also the untreated samples are the topmost microscopy image. Samples were then exposed to the staurosporin to induce apoptosis and subsequently trigger mitochondrial membrane depolarization, and then were stained with TMRM or TMRE. So if you look at the histogram on top, the white peak outlined with black is the depolarized, the cells entering autophagy, as well as the lower microscopy images show the apoptotic cells as well. And you can see there's a drop in fluorescence signal compared to those mock untreated samples. JC1 is another product ICT manufactures for monitoring mitochondrial membrane potential. This is also a lipophilic cationic dye. It has a positive charge in lipophilic solubility, which enables it to readily cross cell membranes, penetrate living cells. Similar to TMRE and TMRM, once inside a healthy cell, JC1 enters the negatively charged mitochondria where it accumulates. JC1 differs from TMRE and TMRM as it accumulates in the negatively charged mitochondria. When it accumulates, it spontaneously forms complexes known as J aggregates, which have an intense red or orange-red fluorescence. The peak emission is somewhere around 595 nanometers. In contrast, when the mitochondrial membrane potential decreases, JC1 disperses into the cytosol, and then it becomes less concentrated, causing it to return back to its monomeric form, which exhibits a distinct green fluorescence property. So JC1, unlike TMRE and TMRM, JC1 has kind of a dual color ratio, allows for a dual color or ratiometric semi-quantitative assessment of mitochondrial polarization states because it can fluoresce orange-red when aggregated in the mitochondria that are polarized, and will fluoresce green when dissipated in the cytoplasm after mitochondrial membranes lose their membrane potential. Here's an image that shows Jurkat cells that were treated with 1 micromolar staurosporin for 2 hours to induce apoptosis, or with DMSO as a negative control, and then were stained with JC1 for 20 minutes. The two cells in the upper and lower left contain mitochondria with polarized inner membranes. Here you can see JC1 is concentrated in the mitochondria. That's evident by the bright orange fluorescence that these cells exhibit. The 3 cells on the right are apoptotic and contain mitochondria that are in various states of permeability. Mitochondria have become depolarized and are no longer able to accumulate the JC1 dye. The cells display an increased green fluorescence. So the mitochondrial membrane potential data I've just shown in this webinar utilize an apoptosis-based model for monitoring changes in mitochondrial membrane potential. And this is because of the link between oxidative stress and apoptosis. That being said, for those of you who are more interested in our mitochondrial membrane potential dyes, I do want to briefly mention the proton gradient uncoupling agent, CCCP. CCCP is a potent uncoupler of the proton gradient, and it acts essentially as a hydrogen ion ionophore, which is a substance that transports ions across the lipid membrane in a cell. The protonated form, which is pictured with the red ionizable proton in the lower right, can diffuse from the intracellular space towards the matrix, helping eliminate the pH differential as it does so. On arrival, it releases this proton to become the lipid-soluble anion form, which then immediately recrosses the membrane, thereby destroying the membrane potential as it does so. This process can then be repeated again and again, and each CCCP molecule can literally do this thousands of times per second. So as you can imagine, this leads to complete dissipation of both the electrical and the pH gradients in the mitochondrial membrane in a very short span of time. I mentioned CCCP just because I would like our listeners to know that this is a chemical included in all of ICT's MitoPT products, making it very easy to generate a positive control using our kit. Now I'd like to switch gears and discuss ICT's products for detecting nitric oxide and nitrosative stress. Nitric oxide is an important physiological regulator of systems such as the cardiovascular system, nervous system, and the immune system. Nitric oxide is made bioavailable through 2 routes. It can be generated through the activity of the enzyme nitric oxide synthase, or it can be ingested in the form of nitrates and nitrites and then rapidly converted into nitric oxide. Nitric oxide is known to have cytotoxic effects, for example, when released by macrophages during an immune response. Additionally, because nitric oxide is highly reactive, it can be converted into a toxic radical responsible for causing nitrosative stress. Now, ICT offers a colorimetric kit for detecting both endogenous nitrite as well as total nitric oxide. This kit has two assay capabilities. The first assay is designed to quantitatively measure endogenous nitrite. During the second assay, nitrate is first converted to nitrite using nitrate reductase, and then is also quantified, resulting in a measurement that correlates to the amount of total nitric oxide. The nitrate concentration can be obtained by then subtracting endogenous nitrite from the total nitric oxide. Our kit has been designed to work with water and buffers, as well as serum and plasma, urine, saliva, and tissue samples. And the data shown on this slide represents a typical standard curve generated using our kit. ICT recently launched a new oxidative stress kit designed for the detection of intracellular nitric oxide synthase activity. This kit utilizes a non-fluorescent cell-permeant reagent called diaminofluorescein 2-diacetate, or DAF2-DA. This reagent is used to detect free nitric oxide and nitric oxide synthase activity in living cells. Once DAF2-DA is inside the cell, cytosolic esterases remove the diacetate groups, releasing DAF2 and sequestering this reagent inside the cell. As once these diacetate groups have been removed, this molecule is no longer membrane permeant, so thus it's retained inside the cell. Production of nitric oxide converts the non-fluorescent dye, DAF2, to its fluorescent triazole derivative, which can also be observed by flow cytometry, fluorescent plate reader, or a fluorescence microscope. This slide shows fluorescence plate reader and flow cytometry results from experiments where Jurkat suspension cells were preloaded with DAF2-DA and then were either mock treated or were exposed to a 1 millimolar DEA nanoate for 30 minutes. This induced NOS activity. If you look at our plate reader results shown on the bar graph, you can see a significant increase in relative fluorescence units when comparing the induced cells and the mock-treated cells. This slide also shows flow cytometry results from induced and mock-treated Jurkat cells. Here you can see a marked shift in mean fluorescence intensity when comparing the mock-treated cells on the top histogram with the induced cells shown in the lower histogram. Approximately 95.5% of the non-08 treated cells stained positive, compared to the mock-treated cells where only 3.3% of the population stained positive. I want to switch topics somewhat to mention some of our products that are geared towards detecting mechanisms for controlling oxidative stress. As I mentioned earlier in this talk, cells use molecules such as antioxidants to help repair the damage caused by oxidative stress. Examples of such molecules include superoxide dismutase, catalase, and glutathione peroxidase. Glutathione is involved in the regulation of many essential biochemical processes in the cell, including DNA synthesis and cell proliferation. Harmful cellular levels of hydrogen peroxide are minimized by the enzyme glutathione peroxidase in a manner that utilizes glutathione as a reductant. The lower image here shows a depiction of oxidation of a glutathione dimer. It's labeled GSSG on this diagram. The oxidation of this glutathione dimer is occurring as glutathione peroxidase converts hydrogen peroxide into two molecules of water. Glutathione reductase converts the glutathione dimer back into two glutathione molecules. So ICT carries a couple glutathione detection kits. These are designed to detect and quantitate both free and oxidized glutathione. Our kits have been validated to work with many sample types, including whole human blood, as well as isolated erythrocytes, serum, plasma, and urine. Our kits are also compatible with most cell lysates and tissue homogenates. This kit utilizes a proprietary molecule that will covalently bind to the free thiol group on glutathione to yield a highly fluorescent or colored product, depending on which kit you pick. Free glutathione is measured first, followed by the addition of a molecule that converts the oxidized glutathione back into free glutathione. A second measurement is taken, allowing the end user to calculate the total amount of both free and oxidized glutathione present in the sample. The data shown here represents a typical standard curve generated when using the fluorescent glutathione detection kit. As I mentioned, we also sell a colorimetric version of this kit. ICT also offers a kit for detecting intracellular levels of glutathione. Our Intracellular GSH assay provides all of the essential reagents for monitoring changes in intracellular glutathione levels by flow cytometry. In this kit, the key reagent is a thiol bright green dye. This proprietary thiol reactive dye quickly penetrates cell membrane structures and accumulates primarily within the cytosol of live cells. In the presence of free thiol-containing molecules such as glutathione, the non-fluorescent thiol bright dye covalently binds to glutathione. This process converts the dye to its green fluorescence-capable form. During periods of oxidative stress, the cytosolic concentrations of the green fluorescent dye become significantly diminished. The reduction in intracellular GSH concentration directly translates into an easily monitored reduction in green fluorescence output within the dying or oxidatively stressed cell population. Glutathione depletion is a common feature of apoptotic cell death, as evident by the flow cytometry results are shown at the bottom of this slide. Here, Jurkat cells were either treated with DMSO as a negative control or were exposed to 1 micromolar staurosporin for 4 hours to induce apoptosis. The effects of staurosporin on the level of intracellular glutathione can be seen in the overlay histograph. The green line represents GSH levels in mock-treated cells, and the red line represents the diminished GSH level in the apoptotic cells. Well, so that wraps up our panel of oxidative stress products. I want to take a quick moment to recap the products I introduced here. The ICT carries products for detecting intracellular ROS activity, hydrogen peroxide, DNA damage in the form of 8-hydroxy-2-deoxyguanosine, mitochondrial membrane potential status, nitric oxide synthase, and glutathione. As you're planning out your project or working through an experiment, please keep in mind that we have a variety of resources available on our website to help you. You can find webinars such as this one on a wide variety of topics. Just go to the archive section on our webinars page. We also have some short product demonstration videos if you would like to see some of our more popular kits in action. We periodically update our blog with helpful tips, new citations, and company announcements, so check there for all your ICT news updates. And finally, we have extensive documentation in the form of product manuals, safety data sheets, certificates of analysis, and so on that are easily accessible from our product pages should you need them. I would like to close by extending a special offer for today's webinar attendees. From now until the end of October, all webinar attendees are invited to take 10% plus free shipping off of the order of any of the oxidative stress kits mentioned in this webinar. And with that, I would be happy to take any questions.
Well, thanks, Dr. Strandberg, for that really informative presentation on oxidative stress. We did have a couple of questions that people chatted in. One of them is, what is the difference between the TMRE, TMRM, and JC1 dyes?
Sure, that can be a tricky topic, and I went over that pretty quickly, so I'm happy to talk about it some more. From a functional standpoint, JC1, TMRM, and TMRE are all very similar. These are all considered cationic lipophilic dyes that accumulate in healthy or polarized mitochondria. Of our MitoPT products, TMRE and TMRM are closely related and are chemically very similar. TMRE is tetramethylrhodamine ethyl ester, and TMRM is tetramethylrhodamine methyl ester. Of these two dyes, TMRE is slightly brighter, having a greater fluorescence potential. TMRM exhibits the lowest mitochondrial binding and electron transport chain inhibition of any of these dyes. But all in all, these two dyes function very, very similarly. JC1 was a little bit more unique in that it exhibits that dual fluorescence property I talked about. In healthy cells with high mitochondrial membrane potential, JC1 spontaneously forms those complexes known as J aggregates. Those are the ones that have that intense orangey red fluorescence. On the other hand, in the apoptotic or unhealthy cells, any cell really with lower mitochondrial membrane potential, JC1 dissipates into the cytosol, and there it can be found in a monomeric form, which shows only green fluorescence. So what this means is, unlike TMRE and TMRM, if you're doing flow cytometry analysis with JC1, you can use a two-color red versus green fluorescence plot. And this allows you to, doing this kind of ratio analysis allows comparative measurements of mitochondrial membrane potential to be made between cell populations.
Okay. Well, thanks, Christy. We've got another question. Can your hydrogen peroxide kit be used to detect changes in intracellular hydrogen peroxide, the H2O2 levels?
Sure, that's an important question. I'm glad that one was asked. The answer is no. This fluorescence-based hydrogen peroxide detection kit format requires the use of a clarified soluble sample matrix, as we found in urine, cell culture media, or buffer, or some kind of buffer-based sample. Whole cell targets would require some type of lysis step. And I do want to mention these kits have not been validated for whole cell hydrogen peroxide analysis.
And another question is, besides the mitochondria, where else inside the cell is ROS generated?
Sure. We talk a lot about the mitochondria because it's a great example, but really there are a lot of different organelles within the cell that can generate reactive oxygen species. Besides the mitochondria, another cellular location of ROS production is the endoplasmic reticulum. This particularly is true during times of ER stress. ROS is also generated in the peroxisomes during metabolization of long-chain fatty acids. It should also be noted that various enzymes, including oxidases and oxygenases, generate ROS as a part of their normal enzymatic reaction cycle.
All right. Well, thanks again, Dr. Strandberg. That's about all the time we have for today. If we were unable to get to your question or your question required a more in-depth technical response, we'll follow up with you personally after the webinar. We'll also be posting a video recording of this webinar in the next day or so, so look out for an email from us after the webinar with a link to the recording, as well as a discount code for some of ICT's products. Our next webinar will be in December, and we'll discuss Alzheimer's disease. Once we have it scheduled, you'll be able to register for it on our webinars page. If you think of any questions after the webinar, you can always send an email to help@immunochemistry.com, or if you have ideas for future webinars, please fill out the Contact Us form on our website or reach out to us through social media on Facebook, LinkedIn, or Twitter. Thanks again for joining us, and have a great day.